Okay. So, good evening, everybody. Welcome to the live interaction session for week six. This session is being conducted at a slightly odd timing at the end of the week. So, I assume there are not a lot of students available for joining the session today. But I'll go ahead and record the video and it will be uploaded for anybody to have access to at any point of time. I don't know why this isn't getting shared. Just okay, somebody joins, then I'll see what needs to be done accordingly. So, okay. Hello, my name is Ankita Bhattacharya. I am a TA for this course, as you've already been watching in the past few live interaction sessions and in the YouTube lectures. So, since this session unfortunately could not be conducted in its regular scheduled time, that's why I have sort of changed the idea a little bit. I will be discussing the week five assignment first of all, and then we'll be following it up with some extensive discussion about immunotechniques that are known to us at this point of time, whatever was discussed in the session, but in more detail with some practical inputs. I have some additional recordings that were part of the work that I do individually in my lab, and I have added that so that it becomes easier to understand for anybody's reference. And then finally, we will be discussing some extra details about the other immunotechniques as well. Which were part of the lecture. And today I will not be discussing the week six sample problems because we will be taking up the assignment tomorrow. So both week six and week seven problems can be discussed together in the session that is to be conducted tomorrow as part of the regular live interaction sessions. I'm very sorry that this could not be conducted before, but I had some other work commitments because of which it was not really possible at that point of time. Just a second. Okay. Okay, so I'll first be discussing some the assignment questions that were part of week five. So if you remember, we had discussed extensively about MHCs and the way in which antigens are presented by class one, class two MHCs, the process in much extensive detail, diseases that are linked with the process, the comparison between MHC presentation by different cell types, the type of pathogens, the different properties of MHCs, all of this. So with this brief recap, let us go over the questions that were part of that assignment. So first of all, we were asked about class 1 MHC genes that encode for membrane-bound glycoproteins are found in A, all nucleated cells, B cells, macrophages, or dendritic cells only B and T cells or tumor cells. So we had discussed this multiple times that all nucleated cells will be able to express class 1 MHCs. Therefore, every cell, every nucleated cell is capable of antigen presentation. But MHC class 2 is present in a much more restricted manner, and cells that are able to present antigens via them are called as professional APCs, which are B cells, macrophages, and dendritic cells. So the correct answer in this case are nucleated cells, which is option A. Second question was about the polymorphicity of class 1 and class 2 MHC proteins and why does it arise. So if you remember, we had discussed the different natures of, and in fact, it was present in the lecture as well, that how exactly you can understand the properties of MHC. So the definition of haplotypes, the way in which they're inherited, how both paternal and maternal antigens together contribute towards maternal and paternal alleles contribute towards the development of the final MHC. So with that, all that in mind, the polymorphic nature came because of two properties, gene conversion and gene recombination. In case you don't remember, you can refer to the slides. I don't think I have that slide here. Yeah, we talked about the fact that genes that reside genes that reside within the MHC region, they are very highly polymorphic. That is because many alternative forms of each gene or allele exists within the population. So that occurs because of gene conversion and because, of course, of gene recombination altogether, giving rise to a huge variety. That means that MHC genes between you and me or any other third person in the population does not necessarily have to have a lot of similarity or a lot of consensus sequences present. It can be quite variable, and in fact, it is, and that is what gives rise to the diversity or the the variation with which in different individuals can respond to different sort of infections. Say, for example, if you talk about COVID, not everybody suffered from COVID because like at the same intensity. Now that depends on two things also: the amount of initial viral titer that was built up in the individual's body. Say, for example, somebody received a very high viral titer, and therefore the virus was able to spread throughout the system of that individual much faster and caused a more severe infection versus another individual who got a less viral load. So that, of course, was different. And that was at that point of time checked using CT values were used for detection via qPCR. Now, while that is definitely one very major contributing factor, something else that also makes a huge difference is the MHC and the immunity of that individual, how strong their immune system is, how quick the response is, and how quickly it is able to eradicate it from the individual's body. So with that in mind, the polymorphicity of MHCs become extremely, extremely relevant. So in this case, the answer is option D, which includes gene conversion and gene recombination both. The third question was about T cells, which can recognize non-self MHC molecules. What are they called as? Self-reactive T cells are those which would be able to recognize own MHC molecules, and therefore those are self-reactive in nature. Alloreactive are those which recognize non-self MHC molecules. Therefore, that is the correct option in this case. T lymphoblasts are different. T lymphoblasts are more primitive, or let's not call it primitive, let's call it a precursor. It is the precursor to the more differentiated, more mature T cells, and therefore they are not the correct option. Therefore, the correct answer in this case were alloreactive T cells, which was option B. Then we are asked about a particular E3 ubiquitin ligase, which is MARCH1, E3 ligase, which is MARCH1, and we were asked to choose correct statements among these three. First, that E3 ligase MARCH1 is expressed in B cells, dendritic cells, and macrophages. It resides in the membrane of a recycling endosomal compartment. It ubiquitinates the cytoplasmic tail of MHC class II molecules. So if you remember the discussion that was made by Professor Shudip Ghosh in his lecture, you would be able to answer this question very easily. That MARCH1 is a E3 ligase, which is involved in maintaining the MHC class II availability on the cell surface. And therefore, all three statements in this case are true. So it is basically the idea for any cell surface receptor in general within cells is the idea of turnover. What does that mean? It means the number of available molecules that are present at any point of time. Say, for example, if there was no turnover, then our entire cell surface would be filled with a major proportion of only one protein. Let's take, for example, insulin receptor. Let's take that. That's one of the most well-studied examples of how insulin receptor is made available on the surface. So for example, if there was no turnover, the insulin receptor would always be present on the surface, and more transcription that keeps on occurring inside the cell and followed by translation would lead to more and more localization of insulin on the surface. So there has to be a checking mechanism, a feedback mechanism, which maintains that this should be the half-life or the sustenance or the turnover of any particular protein at any point of time. So that in case of MHC class II is maintained to a large extent by the E3 ubiquitin ligase MARCH1. And therefore, all three statements in this case are correct, and therefore option D was the correct answer. Then in question 4, we were asked, I think it's question 5, sorry, yeah, question 5, we were asked about MHC class II. It presents antigens that are obtained by phagocytosis, pinocytosis, autophagy, or all of the above. So if you remember the way in which antigen presentation was broadly carried out by class 1 and class 2 MHCs, okay, I cannot find that diagram, just a second. Yeah, so anything that is coming into the cell via pinocytosis, autophagy, or because of the engulfment of anything that was initially extracellular, that is being carried out by MHC class II. So it is capable of presenting antigens that are produced by all three, or that are the result of all three of these processes: phagocytosis, pinocytosis, and autophagy. Therefore, option D was the correct option. I just need to check this for a second. How is the T cell receptor different from the B cell receptor? There are different points of discussion there. So TCR does not contain a variable region like BCR. TCR is never secreted in the plasma. The two chains of TCR are not linked or none of the above. So BCR is basically an antibody. So how does the T cell receptor differ from the B cell receptor? It of course has a variable region, which is giving it the specificity that we talk about. It is never secreted in plasma. I guess that is indeed the correct option. We'll come to it. The two chains of TCR are not linked. It is actually true for B cell receptors as well. While they are B cell receptors have disulfide linkages, which are keeping the two chains intact, both between the heavy chain and light chain, and in between the two heavy light dimers as well. But in case of TCR, the orientation is also consisting of interactions that are linking the two. Therefore, the correct option in this case was that it is never secreted in plasma, while antibodies are definitely membrane bound or secretory in nature. It depends upon what is the how it has been selected and what is the eventual objective to be satisfied using those antibodies. Then finally, we have stimulation of naive cytotoxic CD8 plus T cells into activated cytotoxic CD8 plus T cells. From naive to activated cells is a consequence of cross-presentation of exogenous antigens. Which among the following is the most efficient cross-presenting cell? So cross-presentation was a phenomenon that I don't think I took up in my discussion session, but it was discussed by Professor Shudik. So cross-presentation was best carried out by dendritic cells, which is why they are often considered to be the most potent APCs as well, most useful, most potent, and most diverse set of antigen-presenting cells. Then comes a question which is about if 345 numbers of DRA or DR alpha and 45 numbers of DR beta genes are present in humans, how many types of class 2 MHCs or class 2 HLAs can be expected? So it is a very simple calculation. If you have two entities which can be present in any number, potentially can be present in any number of combinations. So you can expect there to be a multiplication of the two, and that gives you 15,525 possible combinations, all of which can potentially be present on the cell surface. What comes as a question next is whether or not it can sustain. That is a whole different matter to be taken. It might be a productive rearrangement that is allowed to sustain, or it might be something which is self-reactive and therefore needs to be cleared off. Just excuse me for a second. I'm continuously getting a call. Okay. So, yeah. Then comes Then comes the molecular chaperone, which is involved in the assembly of class 1 MHC, whether it is calnexin, HSC73, calreticulin, both A and C, or all of the above. So both calnexin and calreticulin are capable of doing this job. They are involved in the assembly of class 1 MHCs. So option D is the correct option. Finally, for the last question, we were asked about the correct sequence for antigen presentation for any exogenous antigen. So I'm just directly going to the correct option because we have extensively discussed this multiple times before. So it talks about binding of the invariant peptide to the MHC. The invariant peptide gets cleaved. That cleaved peptide is removed and then it allows the binding of the processed antigen in the final step. So correct option is option A. So with this in mind, we will now move to the next discussion. Yeah. So the first set of techniques that we will be discussing today are the immunodiffusion techniques. So what does that mean? It means that it involves movement or localization or change in the motility of antibodies. Immuno is obviously antibody, diffusion means there is some sort of movement which is taking place. So antibodies and soluble antigens interact in an aqueous solution to form soluble lattice, which is called as an antigen antibody complex that eventually develops into an insoluble visible precipitate. So the entire concept of immunodiffusion, which is the first immunotechnique we'll be taking up today, is around the formation of a precipitate, which is when it becomes visible. When something is soluble in a clear, transparent solution, you will not be able to see it or visualize it, right? So both the antibody, sorry, both the antibody and the soluble antigen, both of them are present in solution. For example, let's see here. Um, So here you can see the antigens are present in their soluble format, antibodies are in the soluble condition, and when there is interaction between the two, then at the zone of equivalence, there is formation of a lattice, a soluble lattice that is eventually developing into an insoluble precipitate, and that becomes visible, which is what you see here in a test tube as well. Now, what is happening there? So there are two possible ways in which this precipitation reaction can actually proceed. Either it can be in a polyclonal system or a monoclonal system. So if you do not have a lot of familiarity with this concept, I'll just briefly explain it to you. What does polyclonality and monoclonality mean? So all of the antibodies or a population of antibodies which are able to interact with multiple epitopes of the same antigen would be called as a polyclonal population of antibodies. What does it mean? It means that they are all, say for example, let's take any protein, say let's stick with insulin, right? So insulin as a protein would have hundreds and sorry, would have hundreds and thousands of epitopes on its surface. So any antibody that binds to any part of the insulin protein would be an anti-insulin antibody. But what is to be kept in mind there is that while they are all binding to the same protein, they are binding to different regions. Say, for example, think of this, my hand as a particular protein. An antibody can bind here, something can bind here, something can bind here. They are all binding to the same protein, but different epitopes. These antibodies, while they are anti-insulin antibodies, they are polyclonal in nature. Now, polyclonal antibodies are much easily accessible. They are easier to extract. They are cheaper, obviously. If you are for techniques like immunoblotting, which we'll be discussing just very soon now, in immunoblotting or Western blotting, as it is commonly called, you will find extensive usage of polyclonal antibodies. However, when you talk about monoclonal antibodies, certain experiments necessarily require this population where they bind to exactly the same epitope of one or exactly one epitope of one particular protein. Say, for example, if you see here in the image as well, you will see that all of these antibodies are binding to the blue epitope on this protein. However, polyclonal antibodies are able to bind to multiple different types of epitopes, some in blue, some in yellow, some in the red one, and all of them all together are able to form a lattice, which is forming the precipitate, which is what you are able to visualize. So with this in mind, what you see next is that how does this precipitation or how can this precipitation reaction actually be monitored? So you see a graph that has been plotted where you have antigen added along the x-axis, and what you see on the y-axis is antibody that is being precipitated. So you have three regions. One where there is antibody in excess, which has excess antibody within the supernatant region as well, as is indicated by the plus motif or plus sign. The other extreme is the antigen excess region, where there is more and more or abundance of antigen that is present, which is indicated by this supernatant as well. When you reach an area where they are both not in excess but are present in almost equivalence, then you see that um, there is a formation of this crystal lattice-like structure, which is called as the zone of equivalence, which is otherwise visualized as a precipitate. How does it correspond to the antigen? If you see, you will see that an initial increase, you initially have a population of antibodies, you are gradually increasing the amount of antigen, and you see an increase in the amount of antibody which can be precipitated. At the zone of equivalence, the amount of antibody that can be precipitated or that can be pulled down by the antigen reaches its peak. Beyond that, it exceeds the abundance of antibodies and eventually reaches into an or dips into a region where very less antibody is precipitated, and therefore the curve eventually subsides. So this is the idea, and it is based on the concept that antibodies are able to aggregate certain soluble antigens, and such antibodies are called as precipitants. And the reaction which facilitates this process is called as precipitation. Now we come to the next more details about this. How do you actually visualize this? What you saw here was the formation of a precipitate somewhere somewhat in a soluble state within a tube. When you want to actually visualize this in a different manner, you can do so by looking at precipitation reactions in gels that give you precipitin lines or precipitin rings. These are simple agarose gels through which antibodies are able to migrate, and upon migration they may or may not form a ring or a line of precipitation. We will get into more details about this. When antigens and antibodies they diffuse towards each other in agar, then a precipitin line eventually forms. When antibody is incorporated within the agar matrix, an antigen which is present within the well diffuses into this antibody containing matrix, a precipitin ring will form, and such a reaction is called immunodiffusion. There is a difference between these two. Again, the principle remains the same. The process is quite different. In case of in this case of precipitin line formation, you can see on the left that there are certain sort of caveats made, and that is made using some puncher-like system where you can, punch through the agar and create a sort of little well-like structure. So that ring excised region is forming a well-like structure within which you can load your antigen or your antibody. Once you load both of them, what you will eventually see is, because agar is a porous structure, both antigen and antibody will be able to move towards each other, and upon movement, they are forming this precipitin line-like structure. However, when you are seeing a condition where the antibody is present within the gel and you are incorporating the antigen which is present. So you will just have one hole like this, one or two holes like this, within which you will have your antigen, and within the agar you will have the antibody. Then they are moving towards, or they're sort of diffusing closer to each other, and they're eventually forming a precipitin ring, which is what you see here. These reactions are called as immunodiffusion. But it's not that simple. There are variations within this as well. Immunodiffusion can also be of two different types. So what you see here, there is either double immunodiffusion or there can be radial immunodiffusion. In double immunodiffusion, antibody and antigen are moving towards each other, and what you are seeing at the somewhere in the middle is the formation of this precipitate line. Whereas in radial immunodiffusion, you can see the antibody present within the agar, the antigen present within the well and diffusing towards it, eventually forming the ring. This is the concept I was trying to explain. Now, what exactly happens in that? In SRID, which is single radial immunodiffusion, also called as Mancini's method, an antigen sample is placed within a well and is allowed to radially diffuse into agarose containing a suitable dilution of an antiserum. Antiserum simply means the antibody. So the agarose contains the antibody. And within that, from the well, the antigen sample is gradually allowed to diffuse. As it does so, a region of equivalence is reached. What you see here is a region of equivalence where both of the concentrations of antigen and antibody are coming very close to each other. When they do so, they are resulting in the formation of a presbyterian ring, which is the concentric band that forms around the well. How does it actually or practically look? What you can see here on the right is the image of an actual agar gel within which this has been performed. What you see here are different ratios that have been indicated. So 1 is to 32, 1 is to 16, 1 is to 8, 1 is to 4, and 1 is to 2. These are different dilutions of the antigen which is present, and within the gel you have your antibody. What you can see is that higher the ratio, more is the amount of antigen which is present. You are able to see a much thicker, much more prominent ring. Say for example, for 1 is to 2 or 1 is to 4, you're able to very clearly see a white sort of not very tight, but rather a diffused ring, because the process is occurring by diffusion. It's not occurring within a very sharp, well-defined region, but is instead occurring because of the movement of these two entities towards each other. So what that is eventually doing is that it is forming this ratio. It is forming this precipitation ring or precipitin ring, which is forming some all around the well. So this is how it looks, the single radial immunodiffusion technique. Now, how do you actually use that for quantification purposes? So by comparing the area of the precipitin ring with the standard curve, which is obtained by measuring precipitin areas of known concentrations of the antigen, the unknown concentration of the antigen sample can be determined. So what do you do there? Like you saw here, you have, imagine this, okay, let's take this. Imagine, say, this is your agar gel. Within it, you are punching different sort of holes. And within this agar gel, throughout, you have a certain antibody present. Now, what happens is that through that hole, you have these different dilutions. How do you make these dilutions? Say, for example, the protein is dissolved in antigen is, say, dissolved in either be some sort of liquid, say water, for example, pure molecule water, or high grade double distilled water, basically. Your initial sample has the highest concentration. From there, you are half diluting it to make 1 is to 2. From there, you are making 1 is to 4, 1 is to 8, 1 is to 16, 1 is to 32, so on and so forth. You are diluting the antibody to different extents to see how efficient it is, up to what dilution you are able to get a clear ring. Or just to set up a standard curve. Now, what you'll do for each dilution, for each sample condition, you will measure the area of the precipitin ring. Like for example, here it's way thicker. This is medium thick, this is less thick, this is even less thick, and in the last few cases, you're not really able to see a very clear ring at all. So based on the measurements, you would plot a curve. So on the y-axis, you have the diameter of the presbyterian ring, which is in millimeter, which is in nanometers square, or whatever unit is most suited. And along the x-axis, we have different concentrations. So for each concentration, what is the diameter of the presbyterian ring formed eventually leads to the formation of a straight line curve. And that is your standard curve. This is how protein estimation is actually also done. So that from that standard curve, now what you will do for any unknown protein that you might have, say for example, let's again take insulin. Somebody gives you an insulin sample. You simultaneously have to check what is its concentration and how efficiently it is binding to the antibody that is present. So based on that, you can like commercially also what should be the ideal dilution used for different types of experiments can be predetermined, or at least be largely estimated, with the hope that it will allow for very clean results. So say for example, I get a precipitin ring of 20 nanometers. In that case, I will have to extrapolate from 20, push it back down, somewhere around 25 mg per deciliter of the test reagent is present in the sample that was used. So this is the basic idea of immunodiffusion and its applicability within the lab scenario as well, from a biochemical perspective, to get the unknown concentration of any particular antigen of interest. Now coming to the second type, which is double immunodiffusion or octolony double diffusion or ODD. In ODD, both the antigen and the antibody diffuse from their respective wells towards each other. What does that effectively do? It establishes a concentration gradient. Now, as equivalence is reached, a visible precipitin line would form. We've already discussed this in extensive detail. Now, what is its utility? How is it relevant? Why would we at all go for this technique if radial immune diffusion or SRID was, sustaining the purpose well enough? The idea is that it can be used to estimate the commonality between different types of antigens. Say, for example, let's look at the first case. If there are two antigens which are completely identical to each other, why is that relevant? Say, for example, you have one known sample, one unknown sample, and to identify or characterize that unknown sample, this would be a very, very useful technique if you have a clear hunch in mind in advance. So if two antigens are completely identical, in that case, what you see is that the precipitin line is somewhat V-shaped, like you see here. For an anti-alpha protein, when anti-alpha antibodies are given, then it leads to the formation of this type of a precipitin ring or line. In the second case, when you are adding anti-alpha and anti-beta, in that case, when one antigen is A and the other antigen is AB, because they are partly identical, not entirely, in that case, you still have that precipitin line formation, but its tail is inclined in different directions. In case of this, where they are partially identical, it is inclined towards the left, whereas if it was the other way around, it would be inclined towards the right. And non-identical, which means they are completely different proteins, would result in the formation of this X-shaped line because of the way in which antibodies would migrate. So just what we saw from the image, I'll read it out from this slide. If two antigens share some common epitopes and some non-identical epitopes, then the antiserum forms a line of identity with the common epitopes and a curved spur with the non-identical ones. This is referred to as the partial identity. When they are completely unrelated and they have no epitopes in common, the antiserum eventually will form independent precipitin lines for each of the two antigens. Yeah, that's what you actually see here. That when there are two unrelated antigens, then in that case, they will both keep on trying to interact with each other. And therefore, the line will keep on extending. And that gives you the line for non-identity, which is like a crisscross sort of structure. So with this, immunodiffusion basically comes to an end. You have two different types mainly of immunodiffusion. One was single radial immunodiffusion technique, and the second was Octolodis double diffusion or ODD. So we now come to the second part or second subgroup of today's discussion, which is ELISA. So what is ELISA? That is the first most obvious question. It stands very naturally for enzyme-linked immunosorbent assay. What is it? It is a diagnostic method for quantitatively or semi-quantitatively determining the presence of an antibody or an antigen in a sample in a multi-well format. Typically, a 96 well plate or a 96 well culture dish is used for this assay in particular. So how can you actually do that? You have proteins of interest, or a marker protein or some sort of an antigen of your interest that is adsorbed to the ELISA plate wells. Basically, you link it or you sort of allow it to attach or adsorb, which is the correct word, onto the ELISA plate well. So you basically have a 96 well plate within which you are adding the solution and allowing it to bind. Um, Primary antibodies, which are specific for the protein of interest, are then used to probe the plate wells. After an incubation period, the primary antibody solution can then be removed, and weakly adherent antibodies are washed off by multiple rinses. To detect these bound antibodies, a secondary antibody needs to be added to each well. After an after the incubation period, when you're removing the primary antibody solution, weakly adherent antibodies would be removed. It is very similar to the protocol which is followed for immunoblotting as well. Now, let's go to the figure because I think that will be much easier to understand. So like you see, the first step is for coating of the 96 well plate on the inside using the protein of interest or your marker protein or your target protein. Then in the second step, you are allowing the marker to get well attached to the cell surface. Any site that is not yet bound or not yet attached by any protein, in that case, what will happen? You will block those regions so that any non-specific interactions do not actually end up happening. So you block these unoccupied sites on the well surface with a neutral protein. In most cases, a neutral protein the neutral protein of choice, um, yeah, it is either 2% skimmed milk or 5% skimmed milk. Skimmed milk basically means lyophilized milk powder that is dissolved in TBST, which is the buffer you need for one of the most commonly needed buffers for Western or for any immunotechniques in general. In case of ELISA, what is usually preferred is the use of BSA or bovine serum albumin. What it effectively does is that it blocks any. So I'll just switch on the video for a minute. So yeah, think of this as the plate. Now within this plate, you have some areas where your protein of interest has bound. The rest, okay, this is a better example. So you have these areas where your protein of interest, these 1, 2, 3, 4 regions where your protein of interest has actually bound. The rest of the region is clear. Now what happens is that despite the fact that these plates are not meant to non-specifically bind to anything else, there still lies the potential for any non-specific interaction to take place and to give you falsified results. So what you effectively need to do is you need to block these non-specific areas. And for that, proteins, very rich proteins are used. And most commonly, these proteins are either BSA, like I just mentioned, or it is 2% skim, 5% skimmed milk. 5% skimmed milk is used majorly in immunoblotting or otherwise what we know as Western blotting. Whereas BSA, bovine serum albumin, while it is an expensive alternative. It is much more efficient and is crucial for certain types of proteins as well. Say, for example, when you're working with phosphoproteins, which means which are phosphorylated proteins, they are very abundant in milk and therefore milk cannot be used for blocking in that case. And then in those cases, you certainly need to have BSA as the blocking protein or blocking entity. So while it's an expensive alternative, it is the only option in some cases. So, yeah, with this, we now move on to the next step. So once you have blocked those regions, you are now allowing antibodies or the antiserum to actually bind. Primary antibody is added. It is incubated with the antigen. It is washed to remove any unattached or non-specific antibodies. The secondary antibody, which is used for detection, is then added. And that finally is allowed to bind, then that is washed. So what you're doing is you have a protein of interest. On that, you have your primary antibody. On that, you're having a secondary antibody. This principle is operational for ELISA as well as immunoblotting and very extensively. So it is basically building one on top of the other. I will be coming to that also very soon. But that is the basic idea which is being followed in both of these techniques. It's just that the surface on which it is occurring is very different. In case of ELISA, you have 196 well plate, whereas in case of a 96 well plate or specific ELISA plates are also available. In case of Western, you're doing it on a membrane. So That's the major, and rather I would say the only point of difference. So, with this in mind, we will read the rest of the legend. So, what it says is that typically, in case of the secondary antibody, it's an enzyme conjugated anti-immunoglobulin, and it consists of antibodies against a species-specific portion of the primary antibody, which is used for the testing purpose. So what you're doing is you had this antigen, you had the primary antibody. The Fc region of the primary antibody is bound by the secondary antibody. So it's just looking for, say, for example, primary antibody is being raised in mouse, secondary antibody is only anti-mouse. It will just go and bind to any anti-mouse entity that is present. Or it can be anti-rabbit, it can be anti horse, etc. But the most common, rat as well, for example. But the most common ones are usually these two, mouse and rat, which are, if you remember the lecture, which are most commonly used because A, they are readily available. B, they produce the proteins in much quicker time point. The amount of antigen which is needed to stimulate their or elicit an immune response in their case is much lesser compared to a large animal like, for example, say a horse. So for those reasons, the mouse and rabbit antibodies are the most abundant of the lot, and they are more commercially viable as well. It's more cost effective. Let's put it that way. The final step is the addition of the enzyme substrate, and then you have detection using colorimetric or chemiluminescence-based methods. When the enzyme reaction is finally complete, you get an appropriate amount of A, either the color or the luminescence, and the reaction can be finally halted using a stop solution. So that is how ELISA works. Now talking about the different variants of this. So what you saw in this case was that you were simply allowing antigen antibody interaction. Now, there are multiple variants of this. When the microtiter well plates are coated with samples of unknown antigen concentration, since antigen immobilization is occurring because of non-specific adsorption, it is simply just the adherence of a protein onto a glass-like surface. Therefore, it is important to determine and adjust the total protein concentration as per requirement. After the binding of the antigen is completed, a concentrated solution of non-interacting proteins like bovine serum albumin or casein. When we say casein, it's simply adding skimmed milk, non-fat dry milk, is being added to all of the well plates. This step is known as blocking, like I just explained, because the serum proteins, they block non-specific adsorption of other proteins to the plate. Then serum sample containing primary antibody or primary antibody solution AB1 is added to the antigen coated microtiter wells and is allowed to react with the antigen attached to the well by incubating it for some time at 37 degrees centigrade. So you basically first allow the antigen to bind. Its concentration is not known. You are simply allowing it to bind to the well surface. Then you are washing it, you are blocking it using milk or BSA to all of the plate wells. It inhibits or controls the non-specific absorption or restricts the non-specific adsorption of other proteins to the plate. Then you have your serum sample containing primary antibody, which is AB1, to the antigen-coated microtiter wells, and you allow it to interact with the antigen by incubating at 37 degrees centigrade. After any free antibody or primary antibody is washed away using a wash buffer, the presence of the antibody bound to antigen is detected using secondary antibody. Again, the same principle that okay, so then again, I think this was discussed sometime before, that why are we going through this double incubation step? Why don't we just use a primary antibody which is labeled with some sort of reagent? The answer to this, as I think I might have discussed before, is that if you do that, then A, you are making it a very expensive process. Why? Every single primary antibody that is commercially available would have to be linked to some sort of detection system, visualization or detection system, most commonly chemiluminescence-based detection, HRP conjugation, or some sort of colorimetric conjugation. So that would make it more expensive, and that would also make it more non-specific in nature. So the first binding of antibodies would then only become the final binding, and that is not accurate. When you do it in a stepwise manner, you are refining it and you are filtering out more specific interactions, and therefore your results are more accurate and more reliable in nature. So first, the primary antibody is washed away, then the secondary antibody is added. Any non-specific binding of secondary antibody is added. And finally, the enzyme that is linked to it is used to, the substrate for the enzyme is added. The enzyme degrades that substrate, releases some sort of visualization product, and then that can be quantified. So in colorimetric methods, you go from a colorless substrate to a colored product. This is called a chromogenic substrate. Whereas what you do for a chemiluminescent method is that what you get as readout is luminescence, and then that can be monitored. So the enzyme basically acts as an amplifier. So even if only very few enzyme-like antibodies remain bound, they are very, very capable of generating many, many signal molecules. Say, for example, you might think that there's only one enzyme moiety. It's fine. Enzymes are very, very efficient. So you have a small region with, say, 5 protein molecules to which the antibody is binding. Each of those molecules are capable of degrading almost the entire substrate that is present, and it will give you readout and you will be able to measure it. That's why these techniques are so efficient and so accurate and so extensively used till date. So that is indirect ELISA. So we had direct ELISA, we have indirect ELISA, we have sandwich ELISA next. As the name suggests, it involves two entities within which something else is present. So you have a well which is coated with the antibody. You are adding, so those are called as capture antibodies. These antibodies are capture antibodies to which you are adding your antigen of interest, and wherever is the correct binding site, you will go and bind, which is to be measured. Then you wash off. Every step has to be followed by a washing step for any non-specific interactions to be disrupted. Then you add your second enzyme conjugated secondary antibody, which is the detection antibody. So you have within sandwich ELISA, you have capture antibodies on which protein of interest binds. Then you have the detection antibody, which completes that sandwich, and the detection antibody is linked to some sort of enzyme. That enzyme is able to degrade all of the colorless substrate that is present, and it is able to generate the colored product, which you get as readout. So it is basically just the arrangement or the orientation in this case, which is giving rise to all of this different nomenclature of direct, indirect, or sandwich ELISA. Finally, in competitive ELISA, you have antibody, which is incubated with the antigen that you want to measure. Then what you do is you add this antigen antibody mixture, which is shown here in these antibodies, to this antigen coated well. To that, you are adding the enzyme conjugated secondary antibody, and you are then able to detect the substrate and measure the color. So it basically means you are adding some competition to the existing antigens that are present, and you are able to detect how much of that protein is actually present via the same technique of ELISA that you are measuring the readout. It's just that antibodies are have the major role to play here, and that is why that makes ELISA an immunotechnique and a very, very, very widely used one. Now we'll come to the third type of technique, which is Western blotting, which is something that I very extensively do. So I'm very familiar with the technique, which is where I have also incorporated some of my own like lab videos of how we actually do Western blotting just for reference. It's not for any other purpose. So what does it mean? If you remember the lecture videos, then there, so I had explained that the nomenclature actually comes because of, it starts off from Southern blotting, that Hans Southern had discovered the technique for DNA hybridization based methods. From there came Northern blotting, just because it was completely opposite to the way in which DNA blotting or DNA hybridization was studied. Then from Northern, they came to Western blotting, where it involved the study of proteins. So the goal of Western blotting, also called as immunoblotting, is to identify, using a specific antibody, a particular protein, otherwise known as an antigen, within a complex mixture of proteins. So you have a mixture of proteins. From there, you are detecting or studying the expression or the level or the just the presence of a particular protein. So how do you actually do that? You you have cells. Your starting material are cells or tissues, whatever it may be. From those tissues or cells, you are isolating the entire protein um, compartment. So what is done is the cells are lysed. From there, DNA, RNA is gotten rid of. what remains behind is protein, and that protein remains soluble in some sort of buffer. In my case, in our lab, we work with RIPA or radio immunoprecipitation assay buffer, very commonly used. You can look up the composition if it is interesting to you. You will see that there are different types of ions which are present, which help in keeping the protein stable, which keeps proteases inactive, and which keeps it working for a significant amount of time, and proteins work just fine. So yeah. So that is what is done. You have cells. You are lysing those cells. Lysis simply means that you are uh, that cell is being ruptured and its contents are being released into the environment. And from there, we are only taking out. So the cell has everything, right? It has lipids, proteins, carbohydrates, fats, etc. You are only taking up the protein part of it, and that is a repertoire of hundreds and thousands, 100,000 proteins that are present within the cell. All different possibilities that can be there are present there. And from that population, you are specifically studying one particular antigen of interest using immunoblotting or immunotechnique known as Western blotting. What is the basic outline? First, there is extraction and quantification of protein samples. So like I said, you have these cells. I'll just verbally explain it because you don't need so much detail about the technique. So cells are first washed. So cells, I'll give you an example of cancer cells because that's what I am most familiar with. So basically these cells, they grow on adherent surfaces. You have culture dishes. Like I showed this, say for example, if this was a dish, cells grow attached to a surface. So they are washed first using PBS, chilled PBS, which is phosphate buffered saline, which is just like a buffer solution to clean off any dead cells which might be present on the surface. If there is anything, some dirt or some debris or something unwanted is present, that is washed off. Then these cells are lysed using RIPA buffer, which I just explained. Uh, it contains some detergent, which allows it to actually make pores in the cell membrane and completely allow it to lyse or open up. Then they are scraped well to get rid of or to completely detach any cells and to allow like the RIPA buffer and the cells to interact very, very well with each other. From there, what you get is sonicated. Proteins are sonicated. Different people use different types of cycles that allows the DNA also to get sheared and to completely get rid of that as well. And then finally, from there, you spin it down. You specifically take the protein population which is present suspended within the supernatant, and then that becomes your protein sample for all of the future experiments. Now, what is the amount of protein that is present there is studied using protein estimation. We most commonly, there are multiple ways of actually estimating it, but what we most commonly do are the Lowry method or the Bradford-Lowry method, or many times also BCA assay, which is bisynchronic acid pathway, which is used to detect how much amount of protein is present within your sample, and it is done using by plotting a standard curve. So you have again the same concept. You have different dilutions of a particular protein. With respect to their dilutions, you would expect their absorbance at 540 nanometer to be different. And based on those differences, what you will eventually get is a standard curve. Again, y equal to mx plus c. You can extrapolate any absorbance reading that you get, and you can eventually retrieve the amount of protein which can be estimated. But that is not enough. After estimation, you have to actually separate these proteins based on their electrophoretic mobility, which is their mass to charge ratio. In case of SDS page or polyacrylamide gel electrophoresis containing the SDS detergent, in that case, what happens is there is separation only based on molecular weight because SDS makes all of the proteins negatively charged. If that was not the case, if it was a native page, then some difference in behavior would be observed. Now, the sample is resolved on SES page, transferred onto a membrane support. Don't worry, I'll be showing you images for all of each and every one of these steps when I did it a few days back. Then the addition of antibodies, and finally detection or visualization. Yeah. So what you see here is that first of all, you have the SDS treated protein mix, which is added to each well of the gel. They get denatured in SDS, then they migrate based on their electrophoretic mobility. In this case, it is only the size which is making a difference. Smaller proteins migrate fast. Bigger proteins face retardation or face repulsion, and therefore they migrate much, much, much slower. Then you remove the gel, perform electrode transfer, bind your antigen of interest, and finally add substrate to activate the colored reaction, which is produced as a result. Now here, actually, I have just okay, I'll play the video. This is a very small video to actually show how it works. So this is how you actually make a gel. So these are apparatus which are, so either you can get like pre-casted gels which are available. You can purchase gels. You can have gel casting systems which are like really fast and they're able to cast gels in I think under 2 minutes or something like that. But this is the most traditionally used, most common and most extensively used method, that you have these casting trays. You have two plates, a bigger plate and a smaller plate, which is separated by a spacer. So what you're creating is a small little cavity. Through that cavity, you pour your gel, which has a number of components. You might have learned it by now. So you basically have acrylamide, tris, APS, TEMET, of course, water, and that is mixed well along with SDS as well, because it's a denaturing page or denaturing polyacrylamide gel electrophoresis method. And then you pour it between those two plates and you put that comb, this green colored comb on top to seal it. Now, what does that help? Why are we putting that comb? So from here, this is the casting tray where the gel is initially allowed to solidify or polymerize. After polymerization, this gel is removed. It is placed within this cassette, which can have two gels at a time, which is what you see in the third case, that this is a gel tank. The large structure is a gel tank within which you put your electrophoresis buffer and you place this entire cassette inside it. What you see here are two electrodes at the top, the black and the red electrode at the top, through which that entire system will be completed and will be allowed to run eventually. What you do next is that you have made your gel, cast it, and put it inside buffer. Now you need to put your samples. To do that, first you remove these combs. The comb is extremely relevant in this case, that because it is what is allowing wells to be formed within which you will put your sample. And these combs are also, they are allowing this formation in the correct order, because if there is a say for example, we could have placed something else to make the well, but then their migration would not have been equal, because these are commercially made and each of the wells is of the same side, or each of the uh, teeth of the comb are of the same size, same dimensions, and therefore the wells are made completely uniform or homogeneous in nature, and therefore the migration which occurs from the wells is also very comparable in nature. I'll just zoom out for a second and I'll try to play this video. I think there's some issue. The video is getting lagged. Just a second. Yeah, should work now. So what you see here is that this is my protein sample, which I am loading into the well. I hope you were able to see that. Yeah, I'll play it again. So this you see, what you do is, this is a pipette. It's a micro pipette, which is attached to this sort of tip structure. Yeah. So this is a micropipette attached to a tip. This is a 10 microliter tip within which I have my sample. If I zoom in a little bit, yeah, I think it should be visible. If I zoom in here, you'll be able to see that there are multiple wells which are present. When we remove the comb, 10 such wells have been formed. And within one such well, we are loading our sample of interest. So this is how you load your sample into the well very gently. And this is done for all of the wells. Once that has been done, then you close the top of your gel, and then you see that once electricity starts flowing, the circuit will be completed, and through these, I need my pointer again. Yeah. So I'll play it once more. Yeah, so this is how electricity is flowing through the circuit, and through each of these wells, these proteins will slowly migrate based on the voltage that you have put, voltage or ampere, whatever it is that you vary, that these proteins will slowly migrate through the gel, and they will separate based on how heavy they are or based on their molecular mass. To monitor this, what is also added along with the proteins are ladders. These black colored lanes that you see, they contain ladder, or otherwise also known as molecular weight marker, which is what is used to monitor how far your gel has gone. So what you see here, very faintly, you should be able to see these sort of lines that you can vaguely see, blue and red colored lines. These are actually your protein ladders, which shows that your gel has, my video is off, just a second. So what this effectively shows is that your gel has resolved, which means that proteins that were initially right here, say for example, this is the top side or this is the top side, they have migrated from that top towards the bottom. Once that has happened, what you next do is perform a transfer. So after transfer, um, yeah, after or the arrangement for a transfer involves the construction of this sort of sandwich. That sandwich consists of thick pads, thin pads, membrane. Membranes are commonly either PVDF, polyvinyl difluoride membranes, or nitrocellulose membranes, which are charged in nature. In this case, the membranes that we use are PVDF membranes. They are better for binding a range of different sizes of proteins, very small proteins to very large proteins. It is able to bind them and bind them with good efficiency. So what you effectively do is you can see this sort of 3D structure here as well, that you have this bottom very thick sort of pad, thick pad at the bottom on which you're putting thin pads, on which you're putting the membrane, on which you're putting the gel, again thin pads, thick pads, and then you place it inside a cassette under a lot of pressure so that eventually squeezes and it becomes a sandwich. And each of these components are very heavily pre-wetted, or rather I would say drenched in something called as a transfer buffer, which has methanol, and that helps in conducting electricity through this. Now, by the process of capillary transfer, electrotransfer or capillary transfer, proteins that were present within your gel, if you see this a little carefully, this is a sort of jelly-like substance, right? So proteins which are present in your gel will move from the gel under that electricity pressure, or because of electricity, electrotransfer will allow it to move from the gel onto the membrane. Now the membrane, why are we going into the membrane? The reason for this is that the gel is a porous structure at the end of the day. It cannot be used for blotting. You will not be able to check anything via it. It's just a porous structure. You can stain it using Comasi blue or something like that. But what it effectively will do, it will show you all possible proteins that are present within the cell, because they are just present there. For specific detection, you need a solid support, and that solid support is provided by the membrane. So this is what you see as the 3D structure with all of these layers, one on top of the other. And finally, after transfer has successfully occurred, this is what your membrane should look like. So this is a membrane, PVTF membrane piece. And whatever you saw on the gel has moved on to this solid structure, which is the PVTF membrane, which is, you can see, I can hold it with a forceps. It's a sturdy structure. So now what is next done is that to this PVDF, you add your antibody of interest. Say for example, here I have all of my samples which are currently not visible to me. It has all possible proteins that I have taken from each of my cells. Each of the lanes had different sample conditions. From there, say for example, I want to detect actin. Then I would incubate them. Actin has molecular weight of approximately 45 kD. So these bands indicate what molecular weight I should be looking for. So like for example, this is 250 kD, 120 kD. 91, 71, 54, 43, 33, 29, and finally 16. So these are the different molecular weights that each of these bands correspond to. So if I am to look for something which is around 45 kDa, I will look in this region, which is corresponding to a molecular weight marker of approximately 45 kDa, and I'll probe that region for actin and then see its expression. So that is the whole idea of immunoblotting. And I don't have a result here. Just give me a second. I'll try to share something. Just give me 1 second. My system is okay, no, it's fine. It's facing some issue. Yeah. So my system is sort of crashing a little bit. Yeah, I think it will might work now. Let me just reshare. So what you see here, uh, this is the result of probing the blot that I just showed you. So what I'm seeing, these are all my different samples that is not of relevance to you, but what you see as a readout are the expression of this, is the expression of this protein in all of the different types of samples. So this is what is gotten as a result. So while the entire blot had hundreds of proteins, and the molecular weight markers for 43, 33, and 54 are as shown. And what you get as an output is the protein or the expression of only actin in such a sample. So with this, what you can understand is the specific expression of any protein of interest using the immunoblotting method or using the antibody for your protein of interest in the immunoblotting method. So I hope it was clear. And yeah. Now we'll finally look at a few more things, just a few extra topics to cover up. Uh, Immunofluorescence was the next technique that was discussed. So fluorescent molecules absorb light of one wavelength, which is the excitation wavelength, and they emit light of another wavelength known as the emission wavelength. In immunofluorescence, fluorescent compounds are usually conjugated to the Fc region of an antibody molecule. So you have, like you remember, the enzyme conjugation was performed. Similar to that, fluorescent compounds can also be conjugated. So that does not affect the binding activity of the antibody, but the readout from that antibody can be very effectively or very efficiently studied. If antibody molecules are tagged with a fluorescent dye or fluorochrome, immune complexes containing these can be detected by colored light emission. Antibody molecules bound to antigens within cells or tissue sections can be visualized, and the fluorescence microscope, most commonly known as an epifluorescence microscope, is used in this case, where the exciting radiation is transmitted through the objective lens onto the specimen surface. Once the radiation is absorbed at the appropriate wavelength, it causes the electrons of the fluorochrome to be raised to a higher energy level. When the electrons return to ground state, a characteristic wavelength is emitted, and this results in the formation of a fluorescent image. I hope this is something you guys understand because you might have studied in chemistry, the Jablonski diagram, how it works, what is fluorescence, what is phosphorescence, how they're different from each other, how is it different from luminescence. Um, since there are no other students in class, I'm not able to understand if this is a concept that needs further clarification. So if necessary, maybe we can take it up in tomorrow's session when I'm hoping there would be more students available. Um, but yeah, for now, I think that should be good enough that you have an antibody which is attached to a fluorochrome. Fluorochromes are absorbing radiation at a particular wavelength, emitting at a higher wavelength, and that can be detected as a fluorescence readout. So successful immunofluorescent techniques require the preservation of antigens in forms recognizable by the antibody, suitable antibody, and finally an appropriate label. So with this, yeah. With this, what you need to understand is what are the types of fluorochromes. The most common ones are first fluorescein, which is an organic dye. Most widely used label for immunofluorescence procedures, it absorbs blue light at 490 nanometers and emits a very intense yellow-green fluorescence at 520 nanometers. So what it effectively does is that you have different fluorochromes which are able to absorb and emit light in different ranges. So you can simultaneously stain or detect different proteins. So you have protein A and protein B. Protein A is labeled with its anti-protein A antibody or is detected using anti-protein A antibody, which is linked to fluorescein. You have protein B, which is detected by anti-B antibody attached to, say, for example, rhodamine. So you will get some green fluorescence, you will get some red fluorescence. Some places you'll see a merging of the two, which will be detected as somewhat of a yellowish color. So that is how you'll be able to distinguish each of these different regions. So fluorescein, an organic dye, is the most widely used label, which is absorbing blue light at 490 nanometer, emitting fluorescence at 520. Rhodamine absorbs at 590 and emits at 546. So the incident light will be of different wavelengths in each case. The emitted light will also be of different wavelengths in different cases, and that can also be recorded. And there are softwares which are very extensively used. Now, this is the most basic technique that I'm talking about. A simple microscope, a simple epifluorescence microscope will be able to do this job. There are very, very precise microscopes such as confocal microscopes, which you might be familiar with, which use laser for incidence, and they give extremely crisp images, very high resolution, very crisp images. And there are even more upgrades and updates on this where other more advanced techniques are used to actually get high resolution images with more clarity, perform live imaging using antibodies, monitoring them over time. So all of these, you can make a movie basically out of, say, for example, the movement or the trafficking of a particular protein through the cell. All of these different, very extensively studied processes can do involve the use of antibodies. Um, so as rhodamine emits fluorescence at a longer wavelength than fluorescein, it can be used in two-color immunofluorescence assays by conjugating each to different types of antibodies. So either you can use commercially available antibodies, or what is also done, what I personally do in my lab, is that we have proteins. Again, using the concept of Western only, you have your protein of interest, you have antibody for that protein, you use a secondary antibody for the primary antibody, which is conjugated to something else. So while for Western, you had HRP conjugated or some other enzyme conjugated antibody. In case of immunofluorescence, your secondary antibody is linked to a dye. Either it is FITC labeled, TRITC labeled. These are all different types of dyes. You will see it right here. We'll come to it. Phycoerythrin, rhodamine, etc. All of these different labels. So phycoerythrin is a very efficient absorber of light, very, very efficient, approximately 30 folds more than fluorescein, and a brilliant emitter of red fluorescence. Fluorophores lose their ability to fluoresce as they are illuminated in a process called as photobleaching. Photobleaching basically means that when light is incident for a very long period of time, then fluorochromes are no longer able to emit light of their expected wavelength, and special care needs to be taken to prevent photobleaching through the use of more robust fluorophores that are less bleaching prone, and by increasing their concentration or reducing the intensity or time exposure, light exposure time, etc. So most commonly used for multiple labeling are either AMCA. You can see that each of them have different excitation and emission peaks or maxima, and the emitted color is also different. What you see in the second, what you see as second on the list is actually DAPI. Oh, sorry. Is actually DAPI or 4,6-diamidino-2-phenylindole, which is most commonly used, very, very extensively used to stain the nuclei of cells. So what it does is DAPI binds to DNA or binds to chromatin, and that allows us to detect the nucleus as blue. So in most immunofluorescence imaging experiments or results, you will see a blue colored nucleus, and the cytoplasm or some particular protein is labeled with a different color. So apart from DAPI, Huxt is also another dye which is used. They both do that interchangeably, often used to detect the same thing. Then you have fluorescein, which is FITC, like I just mentioned. You have propydium iodide or PI. You have tetramethylrhodamine or TRITC, which is fluorescing red. FITC is fluorescing green. Indocarbocyanine is fluorescing red. Lysamine rhodamine is fluorescing red. Texas red is fluorescing deep red. And indodicarbocyanine or Cy5 dye, which is the more commercially used name, it gives emission in far red range. Now, far red is outside of the visible range, which you and I as humans will be able to see because the wavelength is longer than the visible spectrum. So in that case, the emission which comes out can be recorded by microscopes, but it will not directly be visible to us. How do I explain this better? Say, for example, all the dyes which fluoresce blue, green, or red, if you look inside the microscope, you will be able to see it. You will be able to see green or red color because it's in the visible range, the emitted light, and you can capture that image. For far red, what it effectively does is I don't know how to explain it very well. Yeah, there are Alexa Floor dyes which do this. Alexa Floor 555, Alexa Floor 488, Alexa Floors beyond 600, 660, etc. So what it effectively does is when you look into the microscope, you won't be able to see anything. But once you capture the image, you can false color it with say pink, purple, or something of that sort. And then that allows you to detect far red range dyes. So that is how fluorochromes can emit in different areas of the visible spectrum or beyond the visible spectrum, and then they can be recorded accordingly. I will end today's class with a very brief recap of what we have learned about FACS. Not going into details, but because it has been already extensively discussed by Professor. And also to keep in mind that since today's class was not interactive at all, I'm very hopeful that we'll be able to do that tomorrow. It was more of a monologue because I kept giving information and information that you might already be familiar with. But yeah, that's the only way I could conduct this class today because otherwise it was not feasible. So anyway, with this in mind, let's just wrap up today's class. I hope that it will be more helpful for the next week. But yeah, and overall, you'll be able to learn some concepts if it is interesting to you. So coming to fluorescence assisted cell sorting. So what is the exact process? You remember the principle. It depends upon the emitted fluorescence, which is used to separate cells or sort cells into different compartments. What does it do? It basically exploits the expression of cell surface markers or proteins which are present at the cell surface and separates them into different groups. So let's take the example shown here. In the example shown, a mixed cell population is stained with two antibodies, one for surface antigen A, other for surface antigen B. Anti-A is labeled with fluorescein, anti-B is labeled with rhodamine. They are loaded into the sample chamber of the cytometer. So this is your beaker containing a mixture of cells with different stains, anti-A or anti-B stains, or in some cases they might be completely unstained because they do not contain the A or B antigen on their surface. This is placed into the sample chamber. So this is a very, very crude drawing of what a cytometer looks like. The flow cytometer looks completely different. You can, just look it up for your reference. It's like a box entity, there are multiple parts of it, sheath fluid passes through it. But basically, there is an area where you can load that sample, and then that sample goes up into the system, and based on the fluidic setup, it is circulated uh, through the tubing of of the instrument, and then sorting can be performed. So the stain cells are loaded into the sample chamber. Cell suspension is expelled in the center of a narrow, rapidly flowing stream of liquid. The flow is such arranged that there is a large separation between cells relative to their diameter. The cells are expelled one at a time from a vibrating nozzle that generates micro droplets containing a single cell. So you have a droplet which contains a cell. Now, laser is passed through this. As it leaves the nozzle, each droplet receives an electrical charge. The computer that controls the flow cytometer is able to detect the emission that comes out of it, and it is able to maintain that each droplet only has one cell. There are setups in the software that are able to detect, because it's a chance event. If the system is controlled in such a way that one droplet should only have one cell, if in case there are droplets which contain two or more cells, then those are called as doublets or triplets, and such doublets can be excluded based on the type of emission that is coming out from them. How is that done? PI staining is often used for that, propydium iodide, which binds to DNA. And then propydium iodide emission can be observed, that if a single cell has X amount of emission of propydium iodide, a doublet will have twice the amount of, or more than twice the amount, and therefore droplets which actually have more than the expected amount, or droplets which emit more than the expected amount of fluorescence should be excluded, because they are not singlets, and therefore they are removed from calculation. And instead what is done is those droplets with a single cell are allowed to pass through, and they are used for detection purposes. The intensity of the fluorescence emitted by each droplet is monitored by a detector and displayed on a computer screen. As it tracks the position of each droplet, it is possible to determine when it will arrive between the deflection planes. By applying a momentary charge when a droplet is passing between them, it is possible to deflect the path of one droplet into another, into one or another collecting vessels. So therefore, what you can do is cells which are, emitting, say for example, anti-A, if it is labeled with fluorescein, for example, which is green in color, cells which are fluorescing only green will be sorted into the population which has which is A positive and B negative, which is shown here. Anything which is any cell or any droplet which is emitting only red, which is only bound by anti-B antibodies, will be sorted into A negative, B positive cell population. Anything which has A and B both will be sorted into this A positive, B positive, or there would be some population which has neither A nor B, and therefore they will be sorted separately. So depending upon the type of instrument that you have, you can sort cells based on their fluorescence into different compartments. So this was a very vague example. What it is effectively eventually used for, I'll give you an example of that. Say for example, you have an entire tissue mass, say any cancerous tissue mass from a surgery or a biopsy that has been performed. From there, you will have a mixture of epithelial cells, luminal cells, etc. You only want to segregate the epithelial cell population from there. So you will use markers for the epithelial cell population and sort it separately, in contrast to the luminal cell markers, which have other distinct cell surface types. So in both cases, the staining would be different, the readout would be different, and based on that, they can be separated. Then you separately want to use the epithelial population for one purpose, the luminal population for another purpose, or maybe it is of no use to you. So that is one example based on which sorting is actually extensively performed. Any change which is monitored on the cell surface, and for which an antibody exists, can actually be used to study and sort using fluorescence assisted or associated cell sorting, or flow cytometry and FACS. So this is the basic idea that I wanted to discuss today. Yeah. So we have reached the end of the discussion for slides, via slides for today. So just to brush up the topics very quickly, and after which I'll be ending the class. The overall focus of this week was to look at immunotechniques. So we extensively looked at the processes which operate for immunodiffusion to be used extensively within the lab environment. Immunodiffusion has different subtypes as well. We understood the concept of precipitation of precipitin ring or line formation for detecting the difference between whether or not antigens are the same, whether they are different, how strong that interaction is, what is the concentration of the antigen that can be detected as well, using certain types of antibody dilutions, or using discrete antibody dilutions, plotting a standard curve and then extrapolating the concentration of the unknown antigen. All of that can be done as well. Following which, we have also looked at the different types of ELISA assays which can be performed. You might be very familiar with ELISA. Uh, like in pathological labs, ELISA is a very, very commonly used technique nowadays. Say, for example, for dengue fever or chikungunya fever, etc. The one of the most common ways in which that detection is done is to look for antibodies for these methods. So it can be direct ELISA, indirect ELISA. So the next time you get some sort of report like this, at your home or somebody you get to know, from there, try to read the assays which are used for this detection purpose, because it will really open up your mind as to how the techniques that you are studying just for some knowledge purpose is being extensively used and is being exploited in different ways for detection of different antibodies or antigens, and commercially are used for a whole other purpose. So all of that can be done. And finally, after ELISA, we looked at its different subtypes, its utility. We did not discuss its limitations, but yeah, of course, it's a very expensive technique. Let me just put it very directly. Um, but yeah, it's used. There is also another technique called as ELISpot, which maybe we'll discuss a little later, because it's actually used for detection of cytokines. So we'll maybe take it up after we discuss cytokines as part of the course, because that might make it a little easier to understand. And after that, we looked at immunofluorescence, that is otherwise also known as ICC, immunocytochemistry. There is also something called as immunohistochemistry, where instead of using cells, you use tissues. Histology sections are used, and they are used to detect certain types of antigens that are present within the tissues. So yeah, that's a different approach, but IHC staining is also very extensively performed. Then finally, we looked at this technique of flow cytometry, which is used. Flow cytometry is the main technique. It's actually, if we directly take the name, then it depends upon the flow, because fluidics is involved. Cytometry means you're measuring something related to cells. So flow cytometry is the major technique, and under that, for analysis, FACS-based analysis is performed, which is fluorescence assisted cell sorting, where you can separate cells into different populations and use them for your own experiment or just for detection purposes. If you want to see the percentage of those cells, if suppose, yeah, okay, we discussed the experiment, so it's fine. So that is the basic idea. So the whole objective of that week was to sort of break the flow of learning about the immune system, the adaptive and the innate immune system, the different branches. But that is what eventually it does boil down to. This was more of a refresher or a breather week. So yeah, that basically sums up all of the topics that I wanted to cover. And we will be taking up the next discussion for T cells, their development, their maturity, how they are able to mediate immunity as well in tomorrow's lecture. So thank you so much for bearing with me throughout this entire journey. Again, I'm extremely sorry for not being able to take last week's class, but I hope at least the lecture uploaded would be helpful. If there are any queries, I am able to see certain comments, but not queries on the YouTube videos. But if there are any queries, please go ahead, write them down in the discussion forum. There are people from the course team who are getting back to you if something directly needs to be communicated to us, to the TAs for the course, the teaching assistants for the course, you can of course bring it up with us as well. So yeah, with that, I think I'll end today's session. Thank you so much. I really hope the course has been helpful and the discussion sessions as well. So we will begin tomorrow with assignment questions and sample problems for the next week as well. And we'll discuss some of the topics with some extra information, some experimental data, etc. So yeah, with that, I will end today's class. Thank you so much and goodbye.